Hello, this is Professor Nassar, and thank you again for joining me for the fourth of 4 lectures covering the gross anatomy of the kidney. This lecture will cover the juxtaglomerular complex of the kidney, otherwise known as the juxtaglomerular apparatus. Again, if you have not gone through the first 3 videos, I'd suggest you do those first, as this region here, we're going to be looking at a very specific portion of the nephron, and having gone through the other 3 videos first, you might find that helpful to provide you with the anatomical context. In addition, if you have your textbook available, I would open it up to the kidney chapter in the book we're using, that's chapter 25, and you should have an illustration that looks somewhat like this, which will allow you to follow along as I'm going through this lecture. In the textbook we're using, that's figure 2510 on page 983. Okay, so let's go through the various parts. Here we're looking at the renal corpuscle, and again, the renal corpuscle consists of 2 parts, the glomerulus and the glomerular capsule. First, let's go through the blood supply to and from the renal corpuscle. We have the afferent arteriole here. That turns into the glomerulus, this capillary tuft that we see here. The glomerulus in this case is exposed here, but enveloped here by another layer of epithelium, which is going to be a visceral layer of the glomerular capsule. We will talk about this in a moment. This is an artificial separation. In this case, the entire glomerulus would normally be covered by this tissue. Here they've stripped that tissue off so that you can see the exposed glomerulus. Here that visceral layer is still on. So all of this is the glomerulus. Here it's covered by a visceral layer. Here it is not. The glomerulus then exits out of the capsule via the efferent arteriole. The efferent arteriole will then go to either the peritubular capillaries or the vasa recta. This is the nephron, and recall the nephron has 2 parts to it. There's the renal corpuscle, and then there's the renal tubule. Note that if you want to impress your anatomy instructor the next time you run into them, it may interest you to know that the renal corpuscle actually has 2 parts. We orient this just like how we orient a globe and we talk about the North Pole and the South Pole. The renal corpuscle has 2 poles to it as well. The part where the blood vessels go in is called the vascular pole, and that's this side of the renal corpuscle. This side, where the filtrate goes out and goes into the proximal convoluted tubule, this side is called the urinary pole. Recall the renal corpuscle has 2 parts. There's the glomerulus here, and then the glomerular capsule, otherwise known as the glomerular capsule, otherwise known as the Bowman's capsule, shown here. Now, keep in mind that the Bowman's capsule or glomerular capsule envelops the glomerulus. The glomerulus is not inside of this space. It is enveloped by the glomerular capsule in the same way that serous membrane will envelop an organ. At this point, you may be spitting out whatever you were drinking because I'm mentioning serous membranes again, and maybe you're sick of hearing about serous membranes. Dr. Nassar, enough with the serous membranes already. Why do we have to know so much about serous membranes, and why do they keep cropping up? Sure enough, they crop up again here. Now, if I could just find, fortunately for me, I talk about this so much, I have a little diagram handy to review. Recall, with serous membranes, your heart, your lungs, your abdominal viscera, they're all surrounded by serous membranes. The serous membrane that surrounds your heart is pericardium, pleura surrounds your lungs, peritoneum surrounds most of your abdominal organs. And again, serous membranes have 2 layers to them. There's an outer layer called the parietal and an inner layer called the visceral. And again, the heart is not in the serous membrane, it gets enveloped by it. So there's an outer layer, an inner layer, and then a cavity or a space in between. Same with the lungs. The lungs gets enveloped by pleura, and there's a parietal pleura, a visceral pleura, and then a pleural cavity in between. Same thing happens here. The one thing to note that is a little odd is that while we have a parietal layer here and a visceral layer here and a space in between, the parietal and visceral layer don't get any specific name. It's not called the parietal nephron and the visceral nephron. It just is called the parietal layer. So the reason I bring this up is if you want to name this after somebody, if you become a famous anatomist and then want to give this an actual name, or name it after a friend or your pet or some other sort of anatomical feature, the opportunity is there. So again, because this is a serous membrane, note the nature of the epithelium. Do we have one layer of cells or multiple layer of cells? One. So this is a simple layer. And what is the nature or the shape of these cells? Are they flat, square, or rectangular? These would be considered flat. So this is a layer of simple squamous epithelial cells. This is the parietal layer. And then that invaginates or turns in on itself to form the visceral layer, shown here surrounding the glomeruli. Recall the visceral and the parietal layer are the same layer. It just turns in on itself, and the visceral layer lies on top of the organ in question. Note that when the parietal layer here moves out, leaves the corpuscle and goes to this region, which is the next part of the nephron, the part of the renal tubule. The first part of the renal tubule shown here is the proximal convoluted tubule. Notice that the nature of the epithelium changes, and now it becomes a layer of simple cuboidal epithelial cells. In this case, the proximal convoluted tubule, the simple cuboidal epithelial cells also have a thin layer of microvilli on their apical ends that face the lumen of the tubule. So we have proximal layer here, visceral layer here. And the visceral layer is basically made up of a cell that's called a podocyte. It's a funny word. Podo means foot and cyte means cell. And so in this case, the visceral layer is not named, but the cells that make up the visceral layer are named, and they are called podocyte cells. And they lie directly on top of these glomeruli. They are called podocytes because they have these foot-like processes that you can see here. These cells almost look like the cellular equivalent of a starfish. There's one here. This is sort of the body of the cell. And then you can see a limb going out this way, an arm going out that way, and so forth. And you can, if you notice, the podocytes, these processes that go out are called foot processes. In some books, the formal term for this is called a pedicel. And these foot processes interdigitate with the foot processes of other cells. You can see here they form this sort of line that resembles the suture of a skull. This line is actually a space through which the fluid in the glomerulus will be pushed out or forced out, and it has as it exits the lumen of your glomerulus and goes into this space, which is called the capsular space. It has to go through this tiny little slit, which is called a filtration slit. It's sort of like if you imagine you take your fingers here, and this would be sort of the foot processes of, I'm going to use my hands as an example. This would be the podocyte. These would be the foot processes of the pedicels of one podocyte. These would be the foot processes of another podocyte, and they basically interdigitate like this. You notice there's a little space there through which you can put through which fluid can pass in and out. That space there is called the filtration slit. The space I'm talking about is this space here through which the pointer is moving through. That's the filtration slit. If you notice, that slit is very, very small, so not everything that's in your glomerulus can pass out through here. The fluid in here, recall, is basically blood, and the blood that's in your glomerulus is blood. And recall, the blood has two components to it. It has a cellular component, and it also has, or what's called formed elements, because there are also non-cellular components like plasma or platelets in there too. And then there's a fluid component of your blood, which is called plasma. The plasma is what we're more interested in this portion of the kidney. The plasma is basically getting forced out of these glomeruli at a rather high pressure, a little higher than your typical capillary. And when the plasma is getting forced out, it basically has to go through a few thin membranes. First, it has to go through the epithelium of this glomerulus. The epithelium is fenestrated, so this is a fenestrated epithelium of the glomerular capillary. Then it has to go through a very thin basement membrane, and then it has to go through the filtration slits of the podocytes. The fluid that passes through here is now in this space here between the parietal and visceral layer, and that's called the capsular space. And the fluid gets renamed now as filtrate. And the filtrate then exits out of the corpuscle, passes through the urinary pole, and then enters into the proximal convoluted tubule. So again, just to remind you, The fluid in the glomerulus is called plasma. As that plasma enters the capsular space and enters the nephron, it's called filtrate. It gets renamed as filtrate. Filtrate and plasma are almost the same thing. Filtrate is basically plasma minus the plasma proteins. Plasma proteins remain in the plasma, at least most of them remain in the plasma because plasma proteins are too big to pass through these filtration slits. Okay, next I want to talk to you about this structure here, which is called the juxtaglomerular complex. Sorry, I don't say that very easily. So each nephron has one juxtaglomerular complex. We call it a complex or an apparatus because it really involves two different components of the nephron that are linked together. The first component is the ascending limb of the nephron loop, where it's about to turn into the distal convoluted tubule. This is the ascending limb here, and we're seeing it in cross-section. The second component is the afferent arteriole and the cells that make up its wall. The book describes, and in the lab we describe a couple of different cell populations that we would like you to be familiar with here. The first are a group of cells that line the ascending limb of the loop of Henle here that are called macula densa cells. And they refer to these sort of tall cells that you see on this side, the right side of your ascending limb, not the cells on the left side over here, which are sort of more normal or more cuboidal in shape. These are a little more columnar. In histologic section, the cells of the macula densa are usually stained very, very darkly. The nuclei are a little more condensed, and they really sort of stick out. And if you notice, the location of this ascending limb of the nephron loop happens to be nestled here right between where the afferent arteriole goes in and the efferent arteriole goes out. This is, we think, deliberate because these two cell populations are going to communicate with one another. What do these macula densa cells do? These macula densa cells help regulate the glomerular filtration rate. Recall that the blood pressure in these glomerular capillaries is relatively high because your body is trying to basically filter your blood. It wants a lot of that plasma in your blood to basically get forced out through these filtration slits and then go into the nephron where the blood can be filtered, or the filtrate can be filtered. And the macula densa cells can basically control the rate at which fluid is moving from the plasma into the filtrate, and that the rate at which it is doing that fluid moves from the glomerulus into the nephron is called the glomerular filtration rate. And you'll learn more about this in lecture. So in these macula densa cells contain chemoreceptors and that monitor or can sense the sodium concentration of your filtrate, and it can also monitor whether the sodium concentration is too high or too low, and it can also monitor if your blood pressure is too high or too low. And if your blood pressure starts to drop, or if your sodium concentration basically starts to fall, if it gets too low, these macula densa cells trigger the release of an enzyme called renin, which get released from your afferent arteriole. The next part of the complex is called, is found here in your afferent arteriole, and the cells that make this up are found here in the wall, and they are called granular cells. They are also known as juxtaglomerular cells or JG cells. They are enlarged smooth muscle cells of the afferent arteriole. And these smooth muscle cells basically act as mechanoreceptors that can sense blood pressure going through the afferent arteriole. So in a sense, they can act as a sphincter that can vasodilate or vasoconstrict the afferent arteriole, in effect controlling the pressure of blood that's going into this glomerulus. And that will then influence your glomerular filtration rate. So if this arteriole basically undergoes vasodilation, then that decreases the resistance to blood flow, and that raises the glomerular filtration rate. These cells also contain secretory granules that contain the enzyme renin. And again, we've talked about renin when we talked about the endocrine portion of this lab. The very first chapter that we looked at in A&P 2 was on the endocrine system, and you may recall renin was a substance that gets released by your kidneys and helps to maintain and regulate blood pressure and sodium homeostasis. And this is where the renin gets released from the afferent arteriole, from secretory granules that are located in the wall. Recall, renin is part of a cascade system. When it gets released, it's part of a cascade system that also includes the molecules angiotensin and aldosterone. And all of these help to regulate blood pressure and sodium homeostasis. Recall, angiotensin 2 raises blood pressure, and aldosterone tells the kidneys to reabsorb sodium. The final concentration of cells that's mentioned in your textbook, I don't think it's mentioned in our lab, so you don't really need to worry about it for this model. And in fact, they're not shown on this model, but are situated right about here in this little nook. And these cells are called extraglomerular mesangial cells. They're located between the arteriole and these cells that make up the ascending limb of the nephron loop. The reason these cells are important is because we think these cells basically pass the signal from the macula densa to the granular cells, when the macula densa basically sends a signal to the granular cells to tell the granular cells to release renin. How does that signal get passed from these cells to these cells? We think the cells in here, these mesangial cells, help to do that. The mesangial cells have gap junctions between them that help relay the signal quickly from one cell population to the other. Like I said, they're not located or shown in this model. They are not mentioned in your lab handout, but they're mentioned in your textbook. And for those who are interested, this is where they would be. And this is one of the functions that we think they hold. Okay, that's it for this demonstration. I hope you enjoyed it. I hope you find it helpful. Good luck in your studying, and we'll see you soon. Bye.